In the second part of this lecture, we're going to cover the capillaries. We'll take a look at how capillaries facilitate the exchange of materials between the extracellular fluids, so that would be plasma and interstitial fluid. Then we're going to take a look at blood velocity. We'll look at veins, and then finally we'll turn and take a look at atherosclerosis, which is a disease of the arteries, and its relationships to strokes and myocardial infarctions. So where we left off was we learned that arterioles control the flow of blood into various capillary beds. Now once blood is in the capillaries, this is going to be where all the exchange happens. So capillaries are designed to facilitate as much exchange between the plasma and the interstitial fluid as possible. Now some components that help maximize the exchange rates are to minimize the distance between those two fluids. So this is done anatomically by making the thinnest wall possible. So this would just be an endothelial lining, no extra wrappings. That would certainly at least facilitate the diffusion of gases. Also helpful for gases is to maximize gradient. So the more oxygen that's in the blood, the more is going to move into the interstitial fluid. That actually is more the job of the respiratory system, so we'll cover that in the third module of this class. And then finally, you want to slow down the velocity in the capillaries. By slowing down the speed of blood as it moves through the capillaries, you give that blood plenty of time to interact with the interstitial fluid. So you could imagine that if blood just zips by really fast, there's not enough time to participate in the exchange. Okay. Now in this image, you'll notice that to minimize the distance between the plasma and the interstitial fluid, we need the smallest layer of cells here. So that's a single layer of simple squamous epithelial tissue wrapped in a cylinder. And of course, that minimizing the distance is going to maximize potentially any diffusion rates between here and here. So now we'll look at three ways that plasma exchanges materials with the interstitial fluid. So they are relying just on simple diffusion. That is done with the gases. So oxygen and CO2 both just move down their concentration gradients via simple diffusion. But the endothelial lining will also participate in endocytosis as shown here. So if you look at this image, you can see that the apical membrane, which is here, is invaginating and grabbing with it stuff from the plasma and then pulling it across and dumping it on the other side. So endocytosis is another way that we can go from plasma to interstitial fluid. Now the other thing to note is that although capillaries come in a variety of different designs, the most common design is called a continuous capillary. So continuous capillaries have the most continuous anatomy, which means the least number of holes in them. But even still, the spaces where the cells join usually will allow some leakage. So that's shown here. We call those clefts. So these are the clefts between the cells of the endothelial lining. And of course, fluid is going to flow down its pressure gradient. So will fluid go from plasma to interstitial fluid or from interstitial fluid back to plasma? It depends. Which one has a higher pressure? If the pressure is higher in the plasma than it is in the interstitial fluid, then fluid is going to get squeezed out of the capillaries and go into the interstitial fluid. The converse is also true. If the pressure in the interstitial fluid is greater than in the plasma, then fluid will move back in to the capillaries. So which one is true? Well, it turns out both of those scenarios are true at different points of the capillary. So this is a little bit of a difficult picture to look at when you first see it, but what this is basically showing is this is the capillary over here that is closest to the arterial. This is the capillary when it's closest to the venule. Now remember, fluid is going to flow down its pressure gradient, but it's the net pressure, overall pressure, of which there can be many different types. And there are two major types of pressure here that are playing a role. One of which is called hydrostatic pressure. Hydrostatic pressure is the physical pressure of blood pushing up against the walls of the capillaries. The other of which is called the osmotic pressure. The osmotic pressure is just the draw of fluid towards an area with more stuff. Now, both of them are going to play a role. Interestingly enough, the osmotic pressure stays relatively constant, and the osmotic pressure is trying to pull fluid back into the capillaries. Why is there more stuff in the capillaries than the interstitial fluid? Because of a thing called plasma proteins, which we'll be covering in the third module. But it turns out that the plasma has more solutes in it than the interstitial fluid does. So consequently, it's always kind of drawing some fluid osmotically into the capillaries. And again, that's a consequence of the plasma proteins. However, we also have the hydrostatic pressure, which is pushing fluid out usually. So this picture right here is showing that when you are closer to the arterioles, the hydrostatic pressure is greater than the osmotic pull, and therefore the net is fluid is going to leave the capillaries and go to the interstitial fluid. In other words, the net is fluid is going to flow out of the capillaries into the interstitial fluid. But as you go down the capillaries, you lose pressure. So this is just part of that graph that we saw last time, where when you look at pressure as a function of vessel, it starts out high in the arteries and then drops, and then it just keeps dropping. So capillaries do this. So the beginning of the capillary is going to have a higher pressure than the end of the capillaries. So as we go down to the end of the capillaries, the hydrostatic pressure ends up becoming less. And now the osmotic pressure takes center stage and starts pulling that fluid back in. Interestingly enough, not all interstitial fluid goes back into the plasma, at least not right away. Some of it gets sucked up into the lymphatic system. So if you've had anatomy, then you know that there's this other system that kind of runs parallel to the cardiovascular system. And it is a series of vessels and organs known as the lymphatic system. Well, the pressure in the lymphatic system is very low, and so fluid naturally is kind of getting constantly sucked up in a low volume into the lymphatic system. Now, the purpose of the lymphatic system is not going to be discussed here, but I do want you to understand this basic overview of fluid movement as we go from plasma to interstitial fluid and then back to plasma or possibly into what is known as lymph. So let's watch that here. In the proximal part of the capillaries, fluid is going to get squeezed out of the capillaries into the interstitial fluid because the hydrostatic pressure is greater than the osmotic pull. Now, as the fluid moves down the capillaries, less and less fluid is going to be squeezed out because the hydrostatic pressure keeps dropping. Now, some of that interstitial fluid will get sucked up into the lymphatic system where it becomes lymph, but the rest of it is going to get recycled into the plasma. So as the blood moves down the capillaries, the hydrostatic pressure is less and less until eventually the osmotic pull is greater than the existing hydrostatic pressure, and that sucks the fluid back into the plasma. Okay, now it's time to talk about blood velocity. I mentioned earlier that blood velocity is going to be slower in the capillaries than anywhere else, which actually should be a little weird to you, because you might think the lowest pressure is actually going to have the lowest velocity. And of course, lowest pressure is found in the veins, not in the capillaries. But it turns out that capillaries have the lowest velocity. Now, from a teleological standpoint, this is a good thing. We want fluid to crawl through the capillaries. By crawling through the capillaries, it gives oxygen and CO2 enough time to reach its diffusion equilibrium. So that's good. Now, why does it have the lowest velocity? Well, you might remember that velocity is a function of the diameter of the vessel. And capillaries have a much, much, much smaller diameter than arteries do. And in fact, this picture that I have up here doesn't even do it justice. The arteries would be like this, and capillaries would be super small. Okay, so arteries clearly have a larger diameter, and capillaries have a much smaller diameter, and therefore fluid should actually be speeding up in the capillaries based on what you told me before, right? What's going on? Okay, yes, if there was only one capillary, that would be true. There are billions of capillaries. And when we take a look at the total cross-sectional area of all of these capillaries put together, we have a total cross-sectional area that is much bigger than the total cross-sectional area of all the arteries. So very similar to a garden drip system, you have a major hose that water runs through, and then you have all these tiny little tubes that come off of it. Well, the more tubes that you have coming off of it, the slower water is going to go through those small tubes, because the total cross-sectional area of those tubes coming off the drip system is actually much greater than the original hose that the water shoots through. So again, the more tubes you add, the slower fluid goes through it, and that's where we get the drip in the drip system. So when we look at the cross-sectional area of all of the capillaries, it has the most. So in other words, you take all your blood and you divide it amongst all these tiny little capillaries here, and therefore the amount of fluid in any one capillary is pretty low, and so it really crawls down. So lowest velocity is going to be found in the capillaries. Okay, so now that we've covered that, let's take a look at veins. This is really an interesting idea. So veins have the lowest pressure, and therefore trying to get blood to come back up to the heart can really be a challenge, especially when you're upright and that fluid has to move against gravity. So veins have one-way valves in them, and many of our veins, which tend to be more superficial on the surface closer to the skin, are positioned in between skeletal muscle so that when the skeletal muscles contract, they actually provide some force. So let me go ahead and diagram that for you here. So the anatomical design of this system involves using both one-way valves and positioning the veins in between skeletal muscles. Now here I have a vein with two valves and some blood that I've highlighted in blue. Now the muscles that are surrounding the vein are going to contract, and when they contract, they're going to bulge outwards. And when they bulge outwards, that's going to put pressure on the veins. So that's actually going to squeeze the veins, much like taking a tube of toothpaste and squeezing that. So once we squeeze the veins, that's going to push fluid up and push fluid down. Now the one-way valves are going to open at the top, allowing fluid through, whereas the valves at the bottom are shut, and so the fluid gets caught there. This forces blood to go upwards only and not downwards. So that means that we have a one-way flow of blood. Now, once the skeletal muscles relax, then what we're left with is more blood at the top area of the veins because we pushed some blood up there. And then there's less blood in the second and third quadrants. So this helps force blood to go one way back towards the heart. Sometimes it is called the muscular pump or the musculoskeletal pump for skeletal muscles. And again, it provides additional pressure that helps move blood against gravity back towards the heart. All right, now that we've covered how this musculoskeletal pump works, where it's actually providing some additional pressure to move the blood, I want you guys to do kind of a cool little experiment. We'll see if this works. Okay, so what I would like you guys to do is I want you guys to stand up and then bend over so that your hands hang low like this. So again, your arms are going to be hanging low, your hands are going to be hanging low, and what you'll see is that your veins pop out. Now, why are your veins popping out? Your veins are popping out because you are having to fight against gravity to move that blood back up. And because there's not a lot of pressure in the veins are going to pop out like you can see here with blood. Now, watch what happens when I do this. Okay, if that worked, okay, you should have seen the veins stop popping out as soon as I took my trunk and lowered it below my hand. Okay, so you can go ahead and try that on your own. And again, what that demonstrates is that the importance of gravity in making it difficult to bring blood back up to the heart, especially again in the lower limbs. So the skeletal muscle pump can help But you have to be moving in order for that to work. When you're just sitting here, it's not doing anything. So the musculoskeletal pump with the one-way valves is meant to assist moving that blood up against gravity. Now, the last thing that I want to talk about with the veins is this idea of why they have smooth muscle around them. So remember, they have smooth muscles so that they can constrict. By constricting, they can help push blood back into the arterial system. Typically at rest, about 50% of your blood is in the systemic veins. That's a lot of blood just sitting there waiting to come back to the heart. Well, let's say I start hemorrhaging. In other words, I'm bleeding a lot. Now suddenly my blood volume gets really low, right? And that extra blood in the venous system is just sitting there. So what the veins will do is constrict so that that extra blood that was sitting in the vein moves over into the arterial system and helps compensate for the blood loss. So anytime somebody has super low blood pressure, is in shock, or it has blood loss or is super dehydrated, you're going to see the venous system constrict as a compensation measure. And basically all you're doing is making your tubes have a lower diameter so you can't put as much blood in them. If you can't put as much blood in them, that means more of the blood is going into the arterial system, where again it's delivering oxygen-rich blood to the cells and nutrients and helping with that, not just sitting here waiting to come back into the heart. Okay. Let's talk about atherosclerosis and its relationship to myocardial infarctions. So atherosclerosis can be summarized as a disease that is characterized by excessive plaque in the arteries. And you can see that pretty clearly on this image. So there's the plaque in a cross-sectional view of an artery. On this one, you can see the plaque there as well. Now first let's briefly talk about what causes the plaque, and then we'll look at the relationship to the myocardial infarctions and strokes actually. So the cause of the plaque is multifaceted, but one relationship that you may have learned about is that individuals who consume chronically high levels of cholesterol are more prone to arterial plaque. So I want to talk about that relationship. When you eat your food, you're eating fats, you're eating cholesterol, you're eating carbohydrates, proteins, nucleic acids. All of that gets broken down in your digestive system and then absorbed into your blood. Well, then what happens? Well, things like glucose and amino acids and nucleic acids are all water soluble. They're hydrophilic, they like water, and so they just float around freely in the blood. But lipids do not like water. So when you try to absorb lipids into your bloodstream, it doesn't want to be anywhere near that. So consequently, you have a number of proteins that are also in your plasma that are meant to kind of carry and protect these lipids in transit. So we call them protein. So these proteins will kind of like wrap themselves around, let's say, cholesterol as it moves through the body. Well, the more cholesterol you consume, the more of these carrier plasma proteins you need. And one such carrier protein is called low-density lipoprotein. For reasons that are not well understood, low-density lipoprotein, when it gets to be in high concentration, likes to wedge itself underneath the endothelial lining into this region of the arteries. Well, your immune system doesn't like that, and so your immune system goes after it and starts kind of breaking stuff down and inducing a bunch of immune responses. So the arterial plaque is basically a collection of both excess lipids, their carrier proteins, the low-density lipoproteins, and some immune cells that have gone in to kind of try to solve the problem but end up exacerbating it. So the outcome of atherosclerosis is that you end up with plaque that greatly reduces the diameter of your artery. So what happens is when we have an artery that's going along and then it narrows because of plaque and now there's a pinch point, is that fluid is now going to have to speed up to go past that. So fluid has to move faster over the plaque. Well, some of that plaque is a little bit unstable. And so much like a pressure washer system, if that fluid moves too fast over the plaque actually gets washed off. So it just bursts and it comes right off. Well, what that does is that exposes the underlying tissue, which should not be exposed to blood, like collagen. Okay, well, you have a set of plasma proteins and platelets in your body that will initiate a clot anytime they see collagen or other things that are underneath the endothelial lining, because to your body, you think that that means that you have a blood vessel that is broken. But instead, no, we've just torn off the top layer, exposing the underlying tissue, and your body develops a clot as a response to that. And the clotting response is initiated upon that exposure. So if you have plaque in the coronary circuit and you blow that plaque off and you develop a clot, that clot is going to move downstream until it basically runs into a vessel that is too small for it to go through. Okay, well, what that means is that no blood can go downstream. So all those arterioles and capillaries that were serviced by that artery lose access to oxygen-rich blood. As we learned about in earlier lectures, the myocardium requires constant and reliable oxygen to work. It does not go anaerobic. It does not function anaerobically. It needs aerobic respiration in order to generate the ATP to keep your heart going. You don't want your heart to fatigue. That would be bad. So without oxygen-rich blood coming its way, the heart muscle dies. Well, interestingly enough, it's not usually the whole heart. It's just whatever's being serviced by that particular artery. The larger the artery that gets blocked, the more extensive the damage is. But if it's a pretty small clot down pretty far into a small artery, it will only be a little bit. And so that's how you can get these gradients of symptoms and severity in terms of myocardial infarction. So myocardial infarction or heart attack is caused by the lodging of a clot somewhere in the coronary circuit that starves downstream tissue from oxygen. That tissue dies. Now, this gives an effect, and one of the things that happens is that tissue goes into rigor mortis, right? Because that's what happens when you lose ATP. But the rest of the heart is still beating, so you get this one dead spot, and the rest of the heart is still beating. Well, that one dead spot, of course, is still connected by anchoring junctions. So even though it's not participating, it's getting pushed, pulled, etc. in all different ways, and now it starts exploding. So the picture that you see here on your left is going to be the heart tissue that has experienced myocardial infarction. And what you can see is that it looks like exploded cells. So this is all debris from inside the cell, junk. You can see that the cells have broken open in many cases. So one of the measurements of myocardial infarctions is to take your blood and measure it for troponin. So just like skeletal muscles, cardiac muscles have troponin in them. Again, that's what's going to regulate the position of tropomyosin and determine whether or not you're going through a twitch or you're at rest. So troponin should never be in the bloodstream. But if you have a bunch of muscle tissue damage and the muscles explode, then troponin gets spilled out everywhere and it gets picked up in the blood and it starts circulating. So not only can elevated troponin levels tell you that someone has experienced a myocardial infarction, you can also get a degree to how serious it is. More troponin equals more damage equals more serious. Okay, so that concludes our lecture. Thanks for putting up with the various conditions.